Hello, and welcome back to another episode of Teach Me in 10. This is, of course, the video series that's brought to you by Technology Networks, where we interview experts and they give you their answers all in less than 10 minutes, making for brilliant bite-sized episodes. And in today's episode, we have the pleasure of speaking with Giovanna Tedesco, who's the head of products at Cresset. And I should think the title already gave this away, but we'll be chatting all about accelerating your small molecule drug discovery projects by using Flair to prioritize the best molecules to make. And before we go any further, this episode is very kindly brought to you by Cresset, who are focused on helping to create a healthier world through trusted state-of-the-art in silico discovery solutions. Research chemists use Cresset software and contract research solutions to discover, design, and optimize the best molecules. And their patented software and expert molecular modelers offer unrivaled insight into protein ligand systems, enabling computational, medicinal, and synthetic chemists to accelerate their research in industry sectors including pharmaceuticals, agrochemical, fine chemicals, and flavors and fragrances. By helping researchers reach better design decisions faster, Cresset Solutions enable them to shorten their project timelines, which can accelerate the time it takes for a product to reach the market. So, without further ado, you're about to watch this incredible Teach Me in 10 in full. Hi Giovanna, how are you doing?
I'm very well, thank you very much.
Fantastic. Well, as we only have 10 minutes on the clock, we best get started. And the first question I really wanted to understand was how Cresset technology transformed new molecule discovery since the company's been formed.
So the history of Cresset starts back in 1996 when our founder, Dr. Andy Winter, published the first paper on the Z-force field for the accurate modeling of ligand electrostatics. Cresset itself was founded in 2002. Since then, we have always worked with our customers and with the Cresset team of discovery experts to deliver new scientific methods to the scientific community, to help them discover, design, and optimize better ligands and accelerate their drug discovery projects. A milestone in Cresset history was the first release of Flair, Cresset platform for ligand-based and structure-based modeling back in 2017.
Perfect. And can you tell us a little bit about Flair and how one might benefit from using it?
Flair is a modeling platform that was created by an expert team of medicinal and computational chemists. In Cresset, in Flair, our users will find robust and well-validated methods for performing day-by-day modeling activities, such as exploring ligand-protein interactions, generating sensible binding poses for their ligands, understanding the SAR of their ligand series, and designing better ligands. At the same time, we also offer a suite of cutting-edge methods that include for gaining insight into protein targets that include molecular dynamics, water stability analysis, and pocket analysis as well. The Cresset implementation of flare-free energy perturbation calculations is also quite popular and will help all users prioritize the best molecules to make thanks to an accurate prediction of binding affinity. However, this is not the only method that we offer for prioritizing the best molecules to make. We actually offer a variety of different methods that go from docking, similarity, electrostatic complementarity scores to Flair FEP scores and predictions, and robust quantitative QSAR models.
Fantastic. So, who would typically use Flair and why?
Flair is used by a growing community of both medicinal and computational chemists. And it is used by pharmaceutical industries, it is used by agrochemical, flavor and fragrances industries, and as well as biotechs and research institutes. We are also very proud of our relationship with academia, and we try to facilitate academic access to Flair by means of a dedicated licensing scheme. At the end of the day, the reason why users use Flair in both the industry and academia is in common to all users. Finding new, better ligands, optimizing these ligands, and accelerating the time it takes to bring new drugs to the market.
That makes sense. So, what would you say are the most commonly used features within Flair? And then could you tell us a little bit about them?
Sure. And docking and scoring is the feature and the method that everybody tries. And in Flair, this is available in a number of different flavors, if you like, that range from normal docking and scoring to ensemble docking, to include multiple conformations of the protein active site into the same experiment, to take into account for active site flexibility, to the docking of covalent inhibitors. Very popular is also molecular dynamics, and we offer a growing choice of different force fields, advanced features, and advanced analysis tools to make modelers' life easier. QSAR modeling is also very popular, and again, we make it available in a number of different flavors that range from field 3D QSAR using Cresset descriptors of electrostatic and shape, to a growing panel of machine learning methods for classification and regression, to qualitative methods of SAR in 2D and 3D. Last but not least, I mentioned it before, Flair implementations of free energy perturbation calculations is also very popular and attracting a lot of attention in both the industry and the academic community.
Nice. And then can you walk us through some of the newer features I know have been included in the latest version 7 release? Because these are pretty exciting.
At each release of the software, we always had new scientific features, more or less all the methods. And the spring release didn't make an exception to this rule, if you like. So we have merged two popular methods for docking, covalent docking and ensemble docking, into ensemble covalent docking, to expand the possibility of including multiple active site conformations in the same docking experiments, also to the docking of covalent inhibitors. For structure-based modelers, a very important new feature is the implementation of GCNCMC, Grand Canonical Non-Equilibrium Candidate Monte Carlo. It is quite a complicated name. This is an evolution, a variant, if you like, of Grand Canonical Monte Carlo methods to improve the efficiency of the exploration of the hydration patterns in the protein active site. The benefit is that when used with the Flair free energy perturbation calculations, it can significantly enhance the predictivity of the method, especially for those proteins, active sites, that are occluded, and they are accordingly more difficult to hydrate. For molecular dynamics, it can also enhance the exploration of the conformational space of the bound ligand. On the ligand-based side of things, we have added a new semi-empirical method to our choice of methods for performing quantum mechanics calculations. This method can be used for the fast geometry optimization of larger ligands and also for the calculation of single point energies. But in Flair, you can also access an optimized workflow in which you can perform a fast minimization of your structures, including larger ligands, followed up with the semi-empirical method, followed by the accurate calculation of energies using one of the ab initio methods available in Flair. For QSAR, we have expanded the choice of available methods with the addition of consensus, regression, and classification.
Fantastic. And then sadly, as we're almost out of time, I just wanted to make sure that I asked what our viewers can do next if they're interested in finding more out about the platform.
Well, they should get in touch with us, obviously. And we would be very happy to organize a free evaluation of a Flair for them and to provide them support with Flair during the evaluation. However, they can also try Flair for free in their own time if they want, because we offer a totally free version of Flair, which is called the Flair Visualizers, that anyone can use. And I would encourage our academic users to get in touch and explore the different options for academic licensing.
Wonderful. Well, thank you so much, Giovanna. It's been an absolute pleasure speaking with you today.
Thank you very much.
Thanks so much for joining us for another incredible episode, and I hope I'll see you again very soon.